some point, I was asking myself, what am I exactly doing here in front of all these brilliant people?
We believe that using these endolysins, we will change the paradigm of the way we fight bacteria.
For the last 2 years, I have been working to design a catalyst. It's not a theory anymore. This is the real deal.
Instead of superficially cutting just the stem of the weed, we're ripping out the whole root.
The concept is fantastic. All their ideas have to be in these 3 minutes.
It's just amazing to see 100 winners, because every talent that has performed today is a winner already.
This is about young scientists. This is about our future in science.
The real problem, ladies and gentlemen, is up here in our minds.
How about recycling human hair into papers? The essence here is to use waste hair as the raw material for paper production.
The effects of the drugs on the bacteria, on the human immune cells, and the pathology in the same environment.
Listening to young people presenting concrete, pragmatic solutions, how to create sustainable food, how to save energy, how to recycle, really gives me a boost for my own work.
People you see have a smile in the face, and I trust them. They will change the world.
And I think each one of them is taking their expertise and truly trying to do something meaningful.
An ecosystem of people who are like-minded.
I think it's definitely about sharing and networking.
The best day in my life.
It's a shame that it's just for 2 days.
Ladies and gentlemen, welcome to the Falling Walls Lab Slovakia 2017, which happens to be the first Falling Walls event organized in our country. My name is Michal Groch and I'm the Master of Ceremony. This event is held under the auspices of the Deputy Prime Minister for Investments and Informatization, Peter Pellegrini. I would especially like to welcome our young scientists and our distinguished guests who are with us today, namely Olga Nachtmanová, the State Secretary of the Ministry of Education, Science, Research and Sport of the Slovak Republic, His Excellency Mr. Joachim Bleicher, Ambassador Extraordinary and Plenipotentiary of the Federal Republic of Germany to Slovakia, Professor Karol Mičieta, Rector of the Comenius University in Bratislava, Professor Pavol Šajgalík, Rector of the President of the Slovak Academy of Sciences, who is not here yet, but as I understand, he is on his way and will be here soon. Then we have Professor Stanislav Hrivňak, Rector of the Technical University of Košice. Professor Tatiana Čorejová, Rector of the University of Žilina, who is apparently also not here, but apparently on her way here. Professor Peter Šimko, Rector of the Slovak Medical University in Bratislava. And Professor and Associate Professor Štefan Rozida, President of the Matador Holding. Also with us, we have Martin Kelle, General Director of Matador. Oh, he's maybe not present yet, also on his way, apparently. And Professor Jan Urban, Chairman of the Humboldt Club in Slovakia. And now, since this beautiful hall is part of the Comenius University premises, and Comenius University also happens to be the main organizer of the event, I would like to give the floor to the rector of the Comenius University, Professor Karol Mičieta.
Dear State Secretary, Excellencies, honorable guests, dear colleagues, ladies and gentlemen. It is a pleasure, great pleasure and honor welcome you at the historical premises to our university on this unique occasion. It seems to be an appropriate combination, tradition and knowledge leading to the future, to the world of innovation. The fall of the Berlin Wall took place 1 week prior to our Velvet Revolution. As a rector of Comenius University, I am proud that students of our university with their colleagues from the schools, other schools in Bratislava demonstrated and called for freedom and democracy even by the prior to Velvet Revolution, and thus that contributed to academic freedom we are enjoying nowadays. And the freedom to research has been the core of our university scientific activity. We witnessed the dynamic changes in the cosmopolitan world we are living in. In addition to migration crises and armed conflicts, the emerging and challenges we are facing today are environment protection, water management, food safety, and many others. The universities definitely have to be involved in searching the appropriate solutions. Innovative approaches are needed, as well the involvement of non-traditional ideas. And today we are welcoming and awarding real innovative leaders, researchers, scientists, and innovators. According to the IT visionary Steve Jobs, innovation is the only way to win. Therefore, actually, all our presenters are already winners. I would like to thank my colleagues and our partners for their enthusiasm and a lot of efforts while preparing this unique opportunity to foster the research and innovation and to create the scientific corridor from Bratislava to Berlin. Let me wish you all of us a successful event. Welcome. Thank you.
Thank you for the welcoming words. And now I'd like to invite here the president of the Falling Walls Slovakia and vice rector for research at the Comenius University in Bratislava, Professor Peter Moco.
Thank you. Before I say anything, I would like to welcome also Professor Robert Redhammer, rector of the Slovak University of Technology. We were not aware that he would come. Ladies and gentlemen, all this is about bright minds and exceptional ideas. I'm very lucky in my life to know many bright people. Many of them are here in this room, and talking to them is always a kind of reward and refreshment. One colleague and friend who always inspires me is Professor Vladimír Bužek. I think that this is the right way to introduce this today's special event. In January this year, Professor Bužek suggested to have a lunch with Professor Jürgen Mlinek, chairman of the board of trustees of the Falling Walls Foundation in Berlin. And after very friendly talking and good meal, I was really convinced that we should bring this attractive event to Slovakia. The idea to organize Falling Walls Lab Slovakia in collaboration of the Comenius University, Slovak University of Technology, Pavol Jozef Šafárik University, Technical University of Košice, University of Žilina, and Slovak Academy of Sciences, was very well accepted and supported by rectors of the universities and president of the academy. The Falling Walls Lab is nothing else than a prestigious worldwide platform on future breakthroughs in science and society. Young scientists, probably many of them the best young scientists, have a unique opportunity to present their groundbreaking ideas in a unique and very attractive competitive format. What is the meaning of the Falling Walls Lab for Slovakia? Our young scientists face a great challenge to directly compete with young scientists from around the world. And I think that this can help them to find their place in a highly competitive European research space. Slovakia will become more visible. The Falling Walls Lab finale in Berlin is attended by hundreds of scientists, decision makers, representatives of top companies and journalists. And finally, ideas really can bring solutions for healthcare, industry, and society. I'm looking forward to all presentations, and I thank you all very much for being here today. Thank you.
And now I would like to give the floor to the State Secretary of the Ministry of Education. Please welcome Ms. Olga Nachtmanová.
Excellency, dear rectors, dear participants, ladies and gentlemen. It's my pleasure to welcome you to Falling Lab Slovakia. I highly appreciate this initiative and its ideas, mainly creation of space for presentation state-of-the-art research projects, initiatives, ideas, and business plans. Young talented people are driving force to development, innovation, and economic growth in Europe. We need to create adequate conditions and instruments and space for creativity, sharing new ideas, experiences, and cooperation with others. Promotion of knowledge, sharing and cooperation between excellent researchers and professionals are main priorities in Ministry of Education, Science, Research and Sport, Slovak Republic. I believe that today's event will contribute to creation of space for new ideas, innovations using in Slovakia and Europe. Thank you.
Falling Walls Lab has originated in Berlin, and Germany has not only contributed significantly to the local round here in Slovakia, but also globally. Please welcome German Ambassador to Slovakia, His Excellency Joachim Bleicher.
Magnificence, Madam State Secretary, dear participants of Falling Walls Slovakia 2017, distinguished guests, ladies and gentlemen. First of all, let me thank you wholeheartedly, those who made this event possible. First of all, Comenius University for lending its premises, the participants for their very good ideas, their respective alma maters for raising these talents, the audience for your interest, the jury for shouldering the burden to determine the winner, all other organizers, sponsors, and promoters for their contribution, the German Academic Exchange Service, the AAD, for covering the cost of traveling to Berlin and accommodation for the winner, and last but not least, Professor Mocho, the president of Falling Walls Lab Slovakia. Dear Professor Mocho, you were convinced the time is ripe for the first Falling Walls Lab in Slovakia. You have worked relentlessly, you have pulled the strings, you have advertised and promoted persistently, and here we are. 21 excellent academics and professionals, 11 men and 10 women, some of the brightest minds of Slovakia, have gathered here at Moyzesovej. You did an amazing job. Thank you very much. And dear participants, in a few minutes, you will be presenting your scientific innovations, your research projects, or your social ideas to this outstanding audience and to a well-selected jury of honorable experts and scientists. Falling Walls offers an amazing opportunity for you and for Slovakia as a scientific location. Today's winners are to participate in the International Falling Walls Final in Berlin. The final winner will get the opportunity to give his or her talk at the Falling Wall Conference on the 9th of November in Berlin. Sometimes the Falling Wall Conference is seen as the most exceptional science conference in the world. 700 international decision makers from politics, business, science, media, culture, from more than 80 nations gather in Berlin to find out which wall falls next, in science or society. Dear participants, I'm deeply impressed by the variety of ideas that we'll be listening to, be it in relation to testosterone or pregnancy, to hydro turbine rotors or flexible robotic cells, cancer treatment, or Einstein's spooky action at a distance, as we could read in the program, to name just a few. You want to break walls, you want to break through, and you want to break free. That is very much the spirit of the peaceful revolution that led to the fall of the Berlin Wall in November '89 and to the fall of the Iron Curtain. The impossible can be made possible if you believe in your idea. Now, it is up to you to convince us of your ideas. I'm keeping my fingers crossed for you and hand back to the Master of Ceremony. Thank you very much for your attention.
Thank you for very inspirational speech. The participants will be evaluated by the jury, led by Professor Vladimír Bužek, representing the Slovak Academy of Sciences. And I would like to invite you to give us a few words about the rules and the evaluation criteria, please.
Okay, dear all, good afternoon. There are many walls to break down, really, in our hearts, in our minds, in the society. And today we have our chance, small chance, to break some of the walls of our ignorance and essentially to build paths to our future with your innovative ideas and sort of research you are doing. So it's really a great pleasure, but also a privilege and responsibility for the members of the jury to listen to all your presentations and essentially to make some judgment at the end, to select the best, if possible. But we will do our best, indeed. Now, what are the rules of the game? But before I tell those rules to you, just a little bit of statistics. There were 56 submissions by the deadline submitted to the Falling Walls Slovakia out of 10 countries, which was really surprising to us that such a response to the call for the Falling Walls Lab Slovakia. So the jury had the responsibility to select of the order of 20 presentations, submissions for today's presentation here in this beautiful hall. Now, we selected 21 projects to be presented. Unfortunately, 3 of the invitees declined the invitation for different reasons, mostly health reasons. So we have today 18 projects to be presented. Now, each project will be presented in 3 minutes, specifically. 2 minutes and 30 seconds for the presentation itself, and 30 seconds for questions. It's very tough. It's extremely tough. All of you who are giving lectures, you know how difficult it is to present your talk. But we want to make your life so hard here in order to prepare you to presentations in your life. Because it's really, usually in life, you have few minutes, 2 minutes to present your greatest idea. And that essentially make a decision in your life. So you have to be extremely clear. You have to be able to present your ideas, the novelty of what you have to say in a very short period of time. And then definitely you have these 30 seconds for discussion. And I believe it will be extremely strict. 2 minutes, 30 seconds. It's the rule of the game. Germans who invented, and they are very pünktlich, so they know exactly that 2 minutes, 30 seconds mean 2 minutes, 30 seconds. Then there will be 30 seconds for discussions. However, because we are more relaxed, so there will be extra 30 seconds. If the discussion is really nice and hot and interesting, then we have this buffer of 30 other seconds. So anyway, so we assume that maximum we will have 3 minutes and 30 seconds for each of the presentations. You multiply by 18 and you know how long this will last. So more or less this is the description. The jury will essentially have to make their marks on the presentation. They might ask you questions during your presentations. And at the end of the day, we essentially, after your presentations, we will have a closed session where we'll compare our notes and we will try to select the 3 winners, the first 3 places, and the winner, number 1, she or he will be essentially, will have a chance to present her or his presentation in Berlin. So more or less this is all. If there are any questions regarding the procedures, ask them now. Otherwise, we probably proceed.
I would just like to introduce other members of the jury. Among them are Dr. Alois Bomba, Associate Professor Peter Celec, Professor Anton Čižmár, Professor Jozef Janovec, Professor Darina Malová, Professor Peter Moco, Associate Professor Štefan Rosina, and Professor Pavol Šajgalík from the Slovak Academy of Sciences, who is still not here yet, but I believe he will take on his job sooner than later. And now, ladies and gentlemen, we are ready to roll with the presentations. Please welcome the first bright young mind, Tomáš Bertok.
Thank you for the introduction. Ladies and gentlemen, I'd like to start talking about prostate cancer diagnostics. This is the 3rd most common cancer in men, with symptoms really similar to benign prostate hyperplasia. Currently, the prostate cancer diagnostics is based on 3 different approaches, one of them being measuring the PSA levels in blood. Currently, the cutoffs used in hospitals are 4.1 nanograms per milliliter and above, with sensitivity only 20%. That means only 20% of prostate cancer patients are actually classified correctly. Going down to 2.6 nanograms per milliliter, the sensitivity goes slightly up at the cost of lower specificity. So basically, any cutoff is a compromise between the assay specificity and sensitivity. And moreover, there is a wide gray zone from 4 to 10 nanograms per milliliter where we are not able to reliably distinguish the BPH and prostate cancer patients. So this approach is about switching from quantitative to qualitative analysis. PSA oncomarker is a glycoprotein with single glycan chain. This glycan changes the composition during the oncogenesis. Currently, glycomics uses heavy machinery such as mass spectrometry. Our approach is based on using lectins, glycan-binding proteins, together with different platforms, electrochemical, optical, acoustic platforms. Even naked eye detection is possible, for example, lateral flow assay, and using different amplification strategies for low-abundant oncomarkers, like enzyme conjugation in common ELISA, or different enrichment methods, for example, using magnetic beads. This last graph is our proof of concept. We are able to distinguish BPH and prostate cancer patients with high selectivity. The sensitivity and specificity were about 18% in each case. And the left graph, as you can see, this is just for comparison, the same groups and the discrimination based purely on their PSA concentration in blood, so you can see almost no difference. That's all. Thank you very much for your attention. If you have any questions, I'll be happy to answer them.
Just a short one. Is your test better than simple digit rectal examination or ultrasound or other methods than biochemistry?
Mm-hmm. The digital rectal exam covers only about 30% of the prostate surface. So basically, the prostate cancer diagnostics must be based on all these three methods.
And our next presenter is Martin Plávala.
So, dear ladies and gentlemen, my name is Martin Plávala. I'm from Slovak Academy of Sciences Mathematical Institute, and this is the wall I would like to break. So the idea is we have two particles, two balls, or we can imagine them whatever, and they are paired, connected, somehow intertwined or entangled if you wish, in a such way that if we ask the first some question, then the second one gives us the opposite answer. This works not only for plus minus questions, but also 1 or 0 or whatever we come up with. And this is very strange. Einstein called it spooky action at a distance. And actually, by definition, I cannot really explain to you how this works. That's by definition. But we actually know what it is. We mathematically can describe it, although it doesn't have a somehow straightforward explanation. And we know how to work with it. What we don't know is why it exists. Why did it happen to be, and why we have this thing. And this is really important to know, because we know that it's useful. We know that we can do very interesting things with it in information technologies and in computation. So if we would know why it exists, maybe we would know more on what to do with it. So how to do this? General probabilistic theories. This is a mathematical abstraction where we not only consider quantum theory as a theory, but we consider every physical theory that could potentially possibly exist in any universe you can come up with. We compare them, we look at their differences, and we apply what we learn to the known theories. In this way, for example, we can describe various concepts such as incompatibility of quantum measurements, which is inherently hard to explain to anyone, based on some black boxes that we can actually construct and present to people. We can come to understand things that before we thought we will never ever understand, just thanks to some mathematical abstractions. And that's everything. Any questions?
So I just wonder, essentially, if, because we are essentially certainly breaking rules or ignorance, but essentially, can you imagine how to test your idea experimentally? What would be the consequence for any of the experiments, needless to say innovations, which I don't think so, but for experimental suggestion of the, essentially you are saying that you are trying to develop new mathematical sort of statistical theories to describe quantum entanglement, but how would you essentially improve the physical understanding of that physical phenomenon?
Well, it's various levels. For example, we can describe it in terms of correlations of non-signaling boxes, which actually shows that we can construct Popescu-Rohlich boxes, and we don't even have to do the experiment because it just follows.
Thank you for your presentation.
Thank you very much.
And we have next coming up Stanislav Hrivňak, please.
Okay, so probably all of you know what is a CT machine. So it's commonly used these days, right, in hospitals, and it's really a great tool for diagnostics of many diseases. But sometimes there is a problem, and that's our wall, that you need to image something that is pretty small, right? So, and you need something that we call microtomography. So, and the question is now, how do we go from macrotomography to microtomography, or CTs? So, for example, you want to image something that is in a millimeter range, that is really small, or maybe even smaller, like small animals or cells or something like that. So there are ways how to perform this, and this is like the basic sketch. How does it look like? So you have some X-rays going out, some sample, and the wave are propagating and so on, and there's some optical element there. And there's detector or some camera to take the image. But the question is there a better optical element to improve micro CTs? And the answer is yes, there is. And so we have developed an optics that is called a Bragg magnifier. It consists, as you see here, of 4 crystals. And the point of these crystals is basically, if you see here, so there's somewhere there the sample, and here's the detector. So as the beam goes, the X-rays, as they go through the crystals, the beam is magnified because of some physics behind it. And because of this, we can use detectors which are larger, and then we can have efficient detection of photons, which is pretty nice because you don't want to kill your sample with X-rays. Okay, and so my contribution to all this project was mainly to build up mathematical model of this optical element and use this to feed it for our developed 3D reconstruction algorithm. And based on this, we have imaged an animal, small animal that is called tardigrade or water bear. It's like, it's big, like hundreds of micrometers with a resolution of hundreds of nanometers. Yeah, so that's it. And if you have questions, I will be happy to answer. Yes?
You said that the resolution is several nanometers.
Yeah.
It's on the range of the X-ray wavelengths.
No, resolution is in the range of hundreds of nanometers, but yeah. So basically you can achieve this by optical microscopy as well, but the thing is that you can't use optical microscopy for optically not transparent objects, but X-ray you can. So like small animals, like beetles and so on.
Now I understand. I had the feeling that you have said that it's in the range of the nanometers, not hundreds.
Yeah, no, hundreds of nanometers, sorry.
Thank you very much for your presentation. It's very interesting. And the next presenter is Štefan Tkáč.
Good afternoon, everybody. My name is Štefan Tkáč. I'm from the Technical University in Košice, and today I'm going to be trying to break the wall of a turbine rotor stabilization. It's a kind of common sense to talk about the efficiency of a hydro turbine. We're talking about the 75% if it comes to microsystems and 96% if it comes to macro systems. The efficiency over here is a crucial issue, but this is exactly something that you might bear in mind in case you are dealing with the worldwide energy production, which is presented in a terawatt hours. So basically 1% is a quite interesting factor. How to deal with that? I'm dealing with the physical connections between the stator and the rotor, and the best way how to deal with that these days is basically by a mechanical system. That means by bearings or by sealants. Right? Another one is by a medium property, which is something that I'm dealing in my project, which is called the MUMT. Basically, it stands for a micro-urban multi-purpose turbine. And in this turbine, I'm applying a floating rotor, which means between the rotor and the stator, there is a small water film, if you understand it, and basically it lifts the rotor gently, and thus there is no physical connection between the stator and the rotor. Another issue is that basically the stator and the rotor, their surfaces are equipped with the so-called slots, which are basically done by a clockwise and anti-clockwise direction, and thus pre-turning the water inside the turbine. And the last, which is the most important part, is the stabilization cone, or conus if you want to put it that way. And basically that one is allowing me to use the spin stabilization, something like in case of a bullet, right? Just I'm dealing with the heavier instances like a turbine rotor, right? At the moment, we have 2 turbines. One is in Taiwan, one is in Slovakia. Both of them are working at the moment. You can apply them as a part of the other turbines that are commonly used. If I'll go back, then we can apply them in a reaction or impulse turbines, or basically we can use it as a turbine on its own. So I think I'm pretty much using the time. If there are any questions, I know it's a mechanical engineering term, so it's a little bit more complicated.
Have you applied for a patent for this idea?
Of course, I did. I have a utility model at the moment.
Will you earn millions or billions for this?
I'm sorry?
Will you earn millions for this?
I hope so. One day.
You get this prize in the Taipei.
Yes,
It's your idea?
It is my patent. It is my idea.
There are no more questions. I would like to thank you for your very interesting presentation.
Thank you very much.
And the next presenter is Terezia Kisková. Please.
So imagine that you come to your doctor once a year for preventive inspection. He would take your blood and urine as always. And then 2 days later he would call you and tell you that your tests indicate that you are in a high risk to become breast cancer patient in few years. Fears, crying, really normal. It's normal feeling. But then it comes just one rational choice: fight for survive. So maybe someone could go to be monitored more frequently to reveal cancer early, or somebody could take some medicaments to like prevention, or somebody could help improve the lifestyle to stay healthy maybe forever. It's just nice illusion, isn't it? But why not let it become the truth? As we know, cancer metabolism differs from that of normal tissue, and because thousands of metabolites are altered during cancer, we need to define them and find out which ones could be relevant to be described as biomarkers, and how do they differ in regard to cancer type. Tumor tissue is forming in the time by the accumulation of somatic mutations leading to selective growth advantage, and then finally to uncontrolled cells proliferation. However, from this time until that time, it is a long time, sometimes decades of years, but it doesn't mean that there happens really nothing, because it's not true. It happens here really something. And I think that there should be one metabolite, or maybe more than one metabolite, maybe hundreds of them, and we just have to define them and find them out. That's why the aim of my project is to show you if the urine metabolomics could be used as a predictive tool in breast cancer initiation process before breast cancer manifestation. Because despite all the efforts, the monitoring of developing stages before manifestation of breast cancer is really impossible in human. That's why we use an animal model of chemically induced breast cancer in rats to see the weekly dynamics, or to see the weekly changes in urine metabolomics, and then we can show if it will really work. Sorry. So we use gas chromatography mass spectrometry, which is actually really fast, easy, and because we use urine, really non-invasive method. And this is actually what we all want. So thank you very much.
Thank you. We still have time for one more question.
Thank you. Does the carcinogen in your experiment affect the metabolites on itself?
Yes.
Okay, thanks.
Thank you. That was very quick. Thank you for your interesting presentation. And the next breakthrough ID will be presented by Patrik Jakabčín.
Ladies and gentlemen, my name is Patrik Jakabčín, and I'm breaking the wall of neoplasia treatment. Okay. Up. Okay. Introduction. 3 out of 100 people in industrial countries have a polyps in gastrointestinal tract. 3 people in these places. It's statistic, okay? Innovative solution. My innovative solution is clear visual border and rise polyps, lasts longer for precise surgical cut, more time for really difficult cases, more than 25 minutes, okay, from 3 minutes to 25 minutes. More than 5 polyps can be removed in gastrointestinal tract within single endoscopy session, okay? From only 1 to more than 5. You can imagine, my right hand is gastrointestinal tract. My left hand is neoplasia, okay? Where is neoplasia? It's a problem. Our solution, the first step, injection. Injection in muscularis propria. After that, elevated neoplastic tissues, and after that, cut. Okay? Let's see the picture. Okay, the time, 0 seconds. Identification neoplasia in gastrointestinal tract. 150 seconds after injection into the muscularis propria and elevated neoplasia. 300 seconds, prepare EMR, endoscopic muconasal resection knife to resection, and 330 seconds, done. Okay? Give me more polyps. Thank you.
You said that 5. 1, it takes 330 seconds, and 5, that means multiply 5?
Sorry?
You have said that you are able, in the reasonable time, to get rid of 5 polyps.
5 polyps.
And you showed
330 seconds.
331.
1, okay.
Times 5?
Times 5, about 25 minutes.
Okay.
All resection, polypectomy.
How this compares to usual?
Sorry?
How this compares to usual approaches? How many, with the standard approach, how many polyps can be removed in 20 minutes?
In 20 minutes, about 6 or 7 polyps. Sorry.
We didn't understand
sorry,
each other.
sorry, okay.
With the usual approach, the standard approach, this is a novel approach
Okay.
you are presenting. But the old approach, how many polyps can be removed?
Only 1.
Only 1 in those
Only
20.
1. Only
so
1.
we are
polyp.
fast. So we are
Yes.
5 times fast.
5 and fast.
Thank you very much. Our next presenter is Barbora Konečná.
Pregnancy complications kill babies and mothers every day. Nobody knows what causes them and why it happens. In most of the cases, no therapies exist yet. The only known fact is that DNA from the fetus, from a child, circulates in the mother's blood during pregnancy. But during pregnancy complications, it happens even more. So we thought, okay, so probably this DNA causes pregnancy complications. I wanted to test this in animal experiment. So I injected mouse fetal DNA into a pregnant mouse. But I wasn't successful. I couldn't induce any pregnancy complications. So I thought, okay, so probably maybe this DNA was just pure DNA, easily edible by degradation enzymes. So probably in real life, this DNA must be hidden from these degradation enzymes. And then I found out about exosomes. Let me explain you what exosomes are. Imagine that I'm a cell of a baby, and you all are cells of a mother. Me, as a cell of a baby, send these small, nano-sized balls to various maternal cells, like this. So these balls are exosomes. They work as messengers between all cells of human organism. They carry DNA, and during pregnancy, they carry DNA of a fetus. They travel from a child to maternal cells. They transfect them. DNA inside of these exosomes is hidden from these degradation enzymes, so they can easily go through. So this is the explanation why our animal experiment didn't work, right? And I want to solve this problem. I want to test this. So if I can prove this, I can solve problems of many pregnant women. Thank you.
You told us, if I can solve it, so do you have indication that you will be able to solve it or not?
Yes. If I had a lot of money, yes. But no. I mean, yes, there is solution. We would need many methods how to characterize all the exosomes that come from a child, because we need to distinguish them from all the other exosomes of all other cells. So all the possible methods would be needed, like proteomics, microscopy, immunogenic methods, and so on.
Okay, so if you suggest this project to Mayo Clinic, will this Mayo Clinic will support you or not?
I think so.
Okay.
Good answer. Thank you very much for your presentation. Thank you. Our next presenter is Diana Drobná.
Good afternoon, ladies and gentlemen. One of the most discussed topics nowadays is obesity. Many people say that obesity is a great threat to our health, but that may be wrong, because people don't die from obesity. People die from complications, such as metabolic syndrome, or also known as metabesity. But what is metabesity? Metabesity is a disease known for its signs. Signs such as high blood pressure, high blood fats, diabetes, and also fatty liver. All of these signs are associated with obesity. The reason for this is inflammation. But what causes this inflammation? We don't know yet. We found out in our previous study that obese people have higher mitochondrial DNA in their plasma. Mitochondrial DNA is found in mitochondria in all of our human cells. What is interesting about mitochondrial DNA is its circular structure. This proves that mitochondria arose from bacteria. Even though this happened a long time ago, mitochondria still have some characteristics of bacteria, which confuses our immunity system, and when there is a higher number of mitochondrial DNA present in our plasma, it reacts as if it was an infection. But this infection is triggered by our own body. So, what if we lower the number of mitochondrial DNA? It would suspend the response of immunity system. We think that discovering treatments for lowering the number of mitochondrial DNA will break the walls of metabesity. Thank you very much.
Do you have any experimental results to support your hypothesis?
We have the experiment that proves that obese people have higher extracellular DNA, and especially mitochondrial DNA. But we don't know yet if the treatment, which would lower the number of mitochondrial DNA, would help the people with metabolic
So
syndrome.
the causation is not clear as yet?
Not yet. It's a hypothesis.
That's a good hypothesis. I am not responsible for mitochondria, that I'm obese.
Thank you very much for your question.
If we do not have any other questions, I would like to thank our presenter. And the next breakthrough idea will be presented by Emese Domonkos. Please.
Dear colleagues, women and men are equal.
but not the same. Gender differences are evident not only in our appearance, but also in the brain functions. As you may maybe noticed, that the prevalence of some psychological disorders, such as depression or anxiety, is higher in women than in men. But what is responsible for these gender differences? We and others have shown that sex hormones, especially testosterone, may be the cause. And dear gentlemen, I guess I will not surprise you, but aging is associated with a progressive decline of testosterone, and low testosterone is causing low libido, erectile dysfunction, but also cognitive and emotional symptoms of aging, such as memory decline and also depression. Testosterone is often given as a supplementation. However, while we know how testosterone affects the prostate and the gonads, the molecular mechanism underlying the effect of testosterone on the brain are unclear. The testosterone is considered to act via an intracellular androgen receptor, modulating gene expression. Due to intensive research on prostate hyperplasia and carcinoma, an alternative mechanism involving membrane receptor have been discovered. Activation of this pathway is much faster and may have a completely different consequences in comparison to the classical pathway. Therefore, we assume that the rapid effect of testosterone on the on several brain functions can be, for example, in the aging man, can be mediated via these membrane receptors. And our previous results show that such receptors are present not only in the prostate, but also in certain brain structures, such as frontal cortex, hippocampus, hypothalamus, or cerebellum. So let us learn about the brain from prostate.
Okay, thank you very much for your presentation. Coming up next is Matej Samos.
Ladies and gentlemen, I would like to talk about tailored direct oral anticoagulation. Direct oral anticoagulants should be preferred today for the prevention of stroke and systemic embolism in patients with atrial fibrillation. However, we have previously demonstrated that despite standard dosage in dabigatran-treated patients, the real plasma concentration might be too low to ensure effective anticoagulation. Moreover, this is a study, recent sub-analysis of the RELY trial, which showed that the incidence of stroke and bleeding in this study correlated with dabigatran plasma levels. Therefore, our research question is, it is possible to improve the clinical outcome of direct oral anticoagulants in patients with atrial fibrillation by monitoring the on-treatment activity and adjustment the therapy. To answer these questions, we have designed a first randomized clinical trial which plans to randomize 300 patients to either tailored DOAC therapy or standard DOAC therapy, and these patients would be monitored for 1 year of clinical follow-up, and we will seek for major ischemic adverse events and also for major bleeding adverse events. In fact, we will try to tailor the therapy either with dose adjustment or with drug adjustments. To summarize, we believe that this study might have ability to answer the question about the role of monitoring of direct oral anticoagulants, and about the role of tailored anticoagulation in clinical practice. Thank you.
Once again, just a short question. Will you measure the drug concentration, or will you measure the physiological parameters for bleeding time and things like that?
Yes, we will measure the drug concentration in one arm of the study. In this arm of the study, we will measure the drug concentration.
Yeah, clear. And how often will you measure it? Wouldn't it be better to do it like in real time, if that would be possible, like during the whole treatment period?
We have a protocol which will measure the therapy at the start of the therapy, 1 month on the therapy, and 3 months after the therapy. Then the drug would be probably, the dosing would be probably sufficient in all the patients in the tailored regimen, and then we will just follow with the clinical follow-up. And the second arm of the study will be without, just fixed dosed regime, without monitoring, without tailoring.
Clear, thanks.
Thank you for your detailed answer and your presentation. And I would like to invite here Lubica Janovičová to present her breakthrough ID.
Good afternoon. So I would like to talk a little bit about DNA. So every living organism is constructed of smaller parts, which are called cells. Inside these cells, we can see DNA. This DNA, however, is also found outside of cells. That's when it's called extracellular DNA. This DNA outside of cells is detected by our immune system and therefore can trigger an immune system response. Extracellular DNA was found to be associated with several diseases, such as rheumatoid arthritis, sepsis, liver and kidney injury. Our studies and studies of others showed that injection of exogenous DNAs improves the state of these conditions and also prevents more tissue damage that already happens there. DNases are such enzymes which are able to break down and break the DNA down to smaller bits. This is also the mechanism which removes DNA from dying cells. So I would like to discuss the other states. So what are the factors which affect the survival and the death of patients? Because not every patient suffering from sepsis dies, and some of them therefore survive. We have a hypothesis that that low DNase activity increases the risk of sorry. We have a hypothesis that low DNase activity increases the risk of extracellular DNA-associated diseases. Simply put, low DNase activity leads to more extracellular DNA and therefore more inflammation. On this picture, you can see gel on which we measure DNase activity. You can see that there is quite a lot of variability. Some of the circles are really small and some are very large. So we think that those small circles could be patients who may not survive after these conditions, and the large circles could be those patients in green who survive these conditions because the extracellular DNA is broken down by these enzymes. So does the high DNase activity protect us from these conditions? We don't know that yet, and we would like to study this variability further, and this is how we want to break the wall of DNA.
Seems like we do not have any questions. Thank you very much for your presentation. And the next presenter will be Matej Mikulášsky, please. Mikulski, sorry.
We live in the 21st century, and the world of computers is bigger than ever. We all use them. And for some of us, they are our professional tool for everyday use. A lot of people like to work with 2 or more applications at the same time, and a lot of these people have only one desktop, or in general, the number of desktops they have is lower than the number of applications they have to work with. And that's where the Great Wall arises. This is the Microsoft PowerPoint with my slides. I was required to use PowerPoint for this talk. And it has the same interface design flow as every other standalone computer application in the world. When we are working with 2 or more applications at the same time, we usually switch between those applications. Or we resize them. We make them smaller, so we can put more of them on a single desktop. And that's where the user interface design flow is. Applications cannot effectively use the space when the size or aspect ratio of the application window is changed. A lot of important control elements, such as buttons, are disappearing when they could have just been relocated. There appears to be a lot of unused space. That is the wall of application user interface design, and we know how to break it. Now, for the first time in the world, I'd like to demonstrate the responsive graphical user interface, or shortly, RGUI. The application shown here will be used for supporting teaching of some advanced chapters of mathematics. Look, you have not seen this before. The user interface changes when the application window is resized. This user interface consists of logical blocks which are always rearranged for providing better user experience. The RGUI uses the space as efficiently as possible, smartly taking into account the size and the aspect ratio of the application window. I'd like to remind you that a similar approach for creating web pages has been proposed only 7 years ago, in 2010, and today it's used all around the world. The responsive graphical user interface breaks the wall in user interface design for standalone computer applications. This approach can be implemented into any computer program right now, starting from today, and it will change the way how we create computer programs and how we use those programs. Thank you very much.
The meaning looks obvious and very useful. So what is the secret why you are the first?
I am asking myself the same question. We have a similar approach for web design for years. It's used everywhere, and nobody thought about the standalone computer applications. Somebody had to be the first.
Okay, so you are more clever than you expected.
Maybe. I don't know.
And our next presenter is Jana Brndiarova, please.
First of all, it's a great honor for me to be here today, and thank you all for coming. I would like to break the wall of the fast cancer detection by graphene. My name is Jana Brndiarova, and I'm at the Slovak Academy of Science at the Institute of Electrical Engineering. So, I would like to detect cancer, in general, by the cancer cell carbonic anhydrase 9, and I'm doing that by the collaboration with the Institute of Virology. The problem is that the standard test, it takes several hours to detect cancer, and they use currently label to detect cancer. Well, so I would like to reduce time of detection, then fabricate label-free cancer detector, and also this detector will be able to detect cancer at the early stage. The solution is the novel 2D material graphene. Firstly, I have to functionalize graphene surface by the antibodies, what is a bioreceptor, to detect antigen, what is a cancer cell. Then the detection will be accomplished by the electrical measurement of the graphene field effect transistor, the changes upon the binding of the antigen and antibodies. Currently, this is proof by the binding of the graphene quantum dots with the molecule Aptos. Here is the result, and this is also the proof of the concept. The same shift, which I can observe by the electrical measurements,
I can observe also by the Raman spectroscopy, which is now a much more easy way, have to distinguish whether there is a binding of the antibody and antigen. So here you can see that this is the area of the monolayer graphene, and here is the shift of the G peak of the Raman spectroscopy, which means that there is something. So there is a binding of the graphene quantum dots to the aptas. So this is a proof of principle, and in the future, this can be useful for a detection of cancer. Thank you, and please, do you have any questions?
I didn't understand why should the use of graphene be better than using cytometry or ELISA or things like that? Have you compared sensitivity, for example?
Yeah, because it's pretty fast. It's faster than the standard normal test. It takes now several hours, and by the graphene, it takes just a few seconds. This is the big advantage of the graphene. It's a novel 2D material. It has a high carrier mobility. This is the big advantage.
In the patent is written that is a carbon.
And I'm sorry, I didn't hear you very well.
Pardon me.
Yeah.
As was said, in the patent name was that the carbon is the important carrier of this detector of the cancer. And you move from the carbon to the graphene, but in between the carbon and graphene, it's also it's graphite, whatever.
Mm-hmm.
What is the benefit of the graphene, from your point of view? I understand the question, the answer that it's quick, but
yeah. Also,
why not something else?
The something else is the early stage detection. Graphene is 2D material, so there is a very good. It has a good ratio between the volume and between the surface. This is a big advantage if I compare it with the
our time
carbon.
is up. Sorry to interrupt you, but we
Thanks.
have to do this and keep it under control. Thank you very much for your presentation. Please. And our next presenter is Mr. Michal Mešťaník, please.
So, dear members of the jury, dear attendants, let me introduce the idea about breaking the wall of stress-induced pediatric hypertension. During recent decades, we have seen the accumulation of evidence that mental stress is one of the major modifiable risk factors for development of elevated blood pressure. Despite this fact, in pediatric patients, the attention was almost explicitly focused only on the effects of obesity, physical inactivity, unhealthy diet, and smoking. So no stress about the stress. Moreover, in these guidelines from the year 2016 on management of high blood pressure in children and adolescents, there is no single mention about mental stress. So the stress was completely omitted. And what's the result? So limiting diagnostics, because stress-related hypertension is considered essential, so we don't have any target which we could treat. Then this results in insufficient or ineffective non-pharmacological therapy, and consequently, many pediatric patients end up with symptomatic pharmacological therapy for the whole life. So this is the wall which we want to break down. And how to do it? So our aim is to complete a diagnostic procedure for stress-related hypertension using a complex psychophysiological test battery, which would allow us to assess the effect of stress and to quantify the effect of mental stress on autonomic nervous regulation and particularly regulation of blood pressure. So what do we want to do is to combine psychological assessment, so questionnaire-related assessment of personality traits, together with together with the modern analysis of biological parameters. So this would allow us to identify the individuals who would benefit from the non-pharmacological interventions, such as increasing the resilience to stress, desensitization of stress response, and facilitation of recovery from the stress situation. And we hope that this strategy could help to pave the way from the symptomatic pharmacological treatment to causative non-pharmacological treatment of pediatric stress-induced hypertension. So actually, the wall which we want to break down is located directly in our brains. Thank you very much.
Are these non-pharmacological treatments evidence-based? Are there any studies that show that really from long term you decrease blood pressure by some of these interventions?
Yeah, so far, there are some studies which indicate that, for example, biofeedback, non-pharmacological treatment could be effective. But the problem is that it's effective, for example, in 40% of the patients. So we want to identify those who had better chance to benefit from those non-pharmacological treatment.
Thank you. Michal Mešťaník. The next presenter is Marcela Nachejová.
Dear ladies and gentlemen, let me tell you something about non-invasive detection of endometrial carcinoma. Endometrial carcinoma is one of the most common malignancies among women in developed countries. Despite this fact, non-invasive diagnostic approaches are lacking. Most endometrial carcinomas developed from precursors in period of years. Many patients who are diagnosed often present clinically with abnormal uterine bleeding, spotting, or discharge many years prior to diagnosis. This offers an opportunity for early diagnosis. Many such patients undergo repeated evaluations such as curettage or biopsy of endometrium. These procedures bring them inconvenience, stress, and the interpretation of the results is limited by adequacy of endometrial sampling. For clinicians, it's a real problem to take samples from extremely obese and polymorbid patients. But there is a still challenge to distinguish minority of women who require management for endometrial carcinoma or its precursors from the others with benign causes of abnormal vaginal bleeding. The very important step is to set a non-invasive collection of samples from all patients. Candidate biomarker studies have identified a number of specific molecular alterations in endometrial carcinoma. Methylated DNA is one of the most promising because its early occurrence in carcinogenesis and its very good stability and detectability. Methylated DNA released from necrotic tumor cells is a very attractive target and has been identified in different biological fluids such as sputum, serum, peritoneal fluid, and also vaginal pool. We can use this fact for non-invasive collection of samples. Patient with using of intravaginal tampons. Patient can self-place the tampon intravaginally. Tampon stay inside for a few hours. After extraction, we are able to detect DNA and also methylated changes from the vaginal pool catched in the tampon. The major point of our study is to include genes that were well documented as methylated in endometrial carcinoma. Non-invasive collection of vaginal pool in combination with sensitive molecular analysis may enable early detection and non-invasive detection of endometrial carcinomas. Thank you for your attention.
Would it be possible that you combine your research with the research on the biosensors and maybe that the tampons would somehow...
Yes, it is possible. It is really possible because we already have some results from fresh endometrial tissue and we would like to apply this in the future on the tampons. We are now working on that. We are collecting the tampons and it is possible to detect DNA and also methylated changes from this DNA from tampons.
Thank you, Marcela, for your presentation. That was very interesting.
Thank you.
Please. Thank you. And we have Maroš Mudrák to present his breakthrough ID.
This is sound of transmission. This is 4 seconds of stamping process. In Slovakia, we are the biggest automotive power. In our country, we produce more than 1 million cars per year. In 3 shifts, 5 single day. And by the way, do you know difference between this car and this one car? in first sight it looks same, but difference is inside this car. Difference is that this one is standard car. It's powered by petrol. We produce 100,000 cars per year from this car. On other side, we've got special car. This special car is powered by CNG. The CNG models, we produce just 18,000 cars per year. So we've got two almost identical cars. These identical cars, we can use tool as you can see in the picture. Cost of this tool is approximately 750,000 euros for one tool. For the explain, CNG model needs put CNG tank, pipes, and grounding pin. So we've got two processes, process of pressing and process of joining. Car manufacturing consists from four operations. First is assembly, first is pressing, body invite, paint shop, and assembly shop. In this world, we've got four different rules, operations, and the processes. With our solution, we are able to provide forming, provide joining, and provide all this stuff and all these processes in one robotics flexible cell. In this cell, we can provide firstly joining, then forming. So this is our solution, and fusion, which fuse will never before operated by any automotive company. My name is Maroš Mudrák. I'm a developer from deepest bottom of my heart, and I came here to break the wall between two worlds of processes of automotive industry. Thank you very much.
Thank you, Maroš. Unfortunately, we don't have time for questions. You have already spent your time for presenting. And please, can you... And finally, we have Ivana Pilchová coming to present her ID. Please.
Good afternoon, dear ladies and gentlemen. Did you know that one healthy cell can produce in 1 minute more than 10 million molecules of ATP? This would not be possible without mitochondria. These are energetic hotspots of cells. Moreover, mitochondria are also involved in regulation of programmed cellular death, and they undergo a strict quality control process. Disruption of mitochondrial homeostasis is a common feature of mitochondrial diseases. Nowadays, the mitochondrial diagnostics is mainly focused on the primary mitochondrial diseases that are relatively rare syndromes. But look at the second group. There you can see more frequent, progressive, neurodegenerative, cardiovascular, or metabolic diseases. Unfortunately, clinicians today only rarely consider them as mitochondrial diseases. And mitochondrial fitness, a potent therapeutic and diagnostic marker, lays behind the wall of neglection. To break this wall, we want to establish a parameter of mitochondrial fitness. Because we believe it's a dynamically responding marker, it depicts the current status of mitochondria, and that could be essentially helpful for patients with a really broad spectrum of diseases. Our unique and innovative diagnostic protocol is based on 3 different experimental approaches. The first one is a functional analysis through high-resolution respirometry for oxygen consumption and ATP production. The second one is molecular analysis revealing mitochondrial DNA copy number ratio and a stage of heteroplasmy. And biochemical analysis reveals us activity of respiratory complexes. All it requires is only 1 blood sample from a patient, and reasonable costs for this examination allows us to perform screenings, multiple examinations of patients to create a temporal prognosis for them. Currently in Slovakia, there is no diagnostical laboratory selectively focused on mitochondrial integrity. We are a newly formed and small research group that want to change it, and my part in this process to bring this idea into reality. Thank you very much.
For my health, are mitochondria in my blood cells as important as in my muscle cells or in my liver and in other tissues?
Yes, they are, but there are still many questions because there are some publications and there are conflicting data because we have publications where were performed measurements on respiratory complexes, but in platelets and other derived blood cells, but there is still question how it can be compared to tissues. But we want to also to check it in our experimental approach, but on a scientific way. This is the diagnostic way. We want to combine it to make it mutually beneficial.
Thank you very much.
Thank you.
And our last presenter is Lucia Stančiaková, please.
And to present you the topic that covers the monitoring of changes in hemostasis and also genetic expression that can be useful for the prediction and prevention of pregnancy complications. We know that there is still some matter in hemostasis that can also contribute to pregnancy complications because physiologically pregnancy means hypercoagulation state. This may be aggravated by the presence of inherited or acquired thrombophilia. So such changes in hemostasis may contribute not only to the recurrent pregnancy loss. As you can see, thrombophilia may be diagnosed in about 40 to 50% of the cases. Then such complications such as intrauterine growth restriction, that is also a complication that is lethal for many pregnancies, or it is the leading contributor to the perinatal and maternal mortality and morbidity. Then the other complications are, for instance, placental abruption, preeclampsia, or for instance, the intrauterine fetal demise. All these complications which we are talking about are the complications derived from the uteroplacental region and circulation that is in such way pathological. So the mean of such prevention of complications is the thromboprophylaxis. Current guidelines contribute to the knowledge of us about means of low molecular weight heparin. It is the drug that is effective and we can use it in pregnancy. But current guidelines also indicate that we still can use them in the whole pregnancy only in the patients who are homozygous for the factor V Leiden mutation and who are with positive family history and past history of some thrombotic or pregnancy complications. All these methods are very demanding, so let me apologize for this. And as you can see, the data which are derived from the current guidelines are based on the retrospective studies which are also performed with the absence of the control group and that are derived from the non-pregnant population. So this is the reason why we try to modify it and also try to investigate the possibility to predict such complications. So there is still the space for the questions we try to answer, and the monitoring of changes in hemostasis may contribute to the better prediction and prevention of such complications, and they are derived from the hemostasis itself. But many patients still come to us after many recovered pregnancies or many pregnancy loss that was also noticed in them, so we try to prevent them. As you have seen, this is the first complex prospective longitudinal study performed all over the world. So we performed the analysis with all the parameters of hemostasis, but still there are some questions that are needed to be asked, such as the genetics, which may contribute to the better prediction of the pregnancy complications. And now we have about 143 healthy mothers and healthy babies.
Run out of time, big time. We cannot continue, even though it sounds very interesting. Thank you very much. And this concludes the presentation part of the event. Please give a big round of applause to all of our 18 speakers. I would like to ask everyone to get seated. We are about to continue. And I would like to welcome the former NASA astronaut Dorothy Metcalf-Lindenberger. Please welcome Dorothy.
Thank you very much for having me here today. It's an honor to be amongst such bright people and people that are thinking forward and how to make our world a better place. So I say congratulations to all of you for participating. The fact that you even put yourself forward and took the risk to stand in front of your peers and present your ideas, that's very noble and to be commended. So I congratulate you, no matter who they have selected to move on to world. It's a big deal that you showed up today. Also, I heard that there was some pressure that you had a countdown clock behind you. So I thought I might take you through my own countdown, which happened on April 5th of 2010. So you can imagine that while everyone else was getting ready for bed on April 4th, my crew was waking up at 8 o'clock p.m. And this is because, of course, when we go to rendezvous with the International Space Station, we have a small window to target that gets us into the correct orbit to rendezvous with the space station. And so we got up at 8 o'clock and we eat a nice light breakfast and say goodbye to our friends within the crew quarters. Then we get suited up into our launch and entry suits. And then finally we walk out and get into this silver van that takes us about 5 kilometers, 6 kilometers out to our launch vehicle. We then get strapped into this vehicle and we wait for 2 hours on our back. And so that is a little bit, a time for trying to relax, but how can you really relax when you're sitting on top of rocket fuel? And also because you've been waiting for this day for a lot of your life. So 2 hours go by, and then finally, the main engines of the space shuttle light. They come up to full throttle and they do a small gimbal, which makes you rock a little bit in your seat. If all is well with the engines, which it was in our case, then the solid rockets light and bam, you are off for a 2.5 minute shaking ride. As you shake, you're trying to monitor, or at least I was trying to monitor different screens and gauges and make sure that this flight is on the right trajectory. After 2.5 minutes, the solids fall off into the ocean. The ride gets a little bit smoother, and we are getting, of course, further and further away from Florida. About 1 minute before all the engines shut off, you start to feel 3 Gs through your chest, and it presses you back into your seat. And then finally, when you feel like you can't take it anymore, the engines shut off, and you're in space. Now, I tell you this because all of our science equipment has to go through the same launch to get to space, which means that many researchers have made sure that it can endure the shaking as well as the radiation and all of these things that get their research into space. And I really wanted to talk to you more about the research that's going on in space because you were talking about what you're thinking about doing with your research. And I noticed that a lot of today's presentations were on medicine and the human body and how we understand the human body, as well as, of course, technology and physics. So I thought I'd start out with some pretty impressive things that have been going on board on the International Space Station. And these things are going on so that we can break down the walls of lower Earth orbit and get on to exploring Mars. And so one of the major things accomplished last summer by Dr. Kate Rubins was to finally sequence DNA in space. It really was only doing the sequencing. All the preparation was done on Earth to really just look at, could we sequence DNA in space? And looking at the technology of the instrument to do it and to see if it could withstand all of this shaking and the trip to space. And it did, and it worked out well. And this is great because, of course, it can tell us more about the human body as it travels in space, as well as be able to identify possibly new life on Mars. So we'll continue to have DNA sequencing in space, and we'll use Earth as we have some folks that study underwater and use this analog to do the whole preparation and practice before we send these procedures into space. So we have sequenced DNA in space and made that great contribution. Furthermore, I recognize that a lot of people had been doing work with cancer and cancer research, which is also very critical for space exploration. And we have had over 40 years of thinking about humans facing all this radiation in space, as well as different challenges in space, might have a higher rate of cancer. Some things we've learned is that by interrupting the circadian rhythm, just like the circadian rhythm is interrupted for astronauts in space, may cause higher rates of cancer, not just the radiation, but the actual changing of the circadian rhythm. And so that has led to light therapy back on Earth. And new ways of treating cancer can be better researched in space because in microgravity, the cancer cells can develop into large 3D organisms versus more of the flat structures that develop here on Earth. So that's led to better treatment of prostate cancer through microencapsulation and directly treating the cancer versus having to do a lot of chemotherapy and treating the whole body. And more research, of course, is going on in immunotherapy. So I think that's very positive outcomes as well. Near and dear to my heart are the earth sciences. My background is in geology. And humans are not as involved in the earth science research. It's more the type of technology we use to observe the Earth. But of course, we're sending sensors up to better understand the aerosols that are in our atmosphere, also to measure lightning and the contribution of rainstorms down in the tropics and understand if they're changing or how they're maybe remaining the same, so gathering data there. As well as, of course, understanding carbon dioxide and its movement throughout the atmosphere. So those are some of the developments in the earth sciences. We've had the International Space Station on orbit for over 17 years, and engineering is something that we sort of take for granted, but this has been a fantastic engineering feat done by multiple countries, including the European Space Agency. We've continued to learn how to do better spacewalks and to repair equipment that is aging and breaks down, and we've learned how to build and make hardened equipment and quite frankly, just how to repair this equipment so that we can go on and build better structures that would be lasting on Mars. And then we've been able to understand more about the cosmos. Again, this is not so much interactive with humans, it's research that comes back to Earth, but we have the alpha magnetic spectrometer on board the station. It's been there since 2011, and of course this is helping us look for dark matter or antimatter and helping us understand our place in our universe. And so the International Space Station, like I said, is a place for all countries. It is truly international. There's now a new aspect where research can be flown for corporations as well as academia, and that's handled through our CASIS, which is a separate entity. It's not just NASA or the European Space Agency. So if you have great ideas that you would like to see in space, think about entering them there as well. And with that, I just want to say again, thank you for the research that you're doing, for pushing the boundaries and breaking down the walls. Like I said, we can only get forward, we can only get on to Mars if we think about how to do this together and to do the research and work together. It was my pleasure to fly in space. I didn't personally get to do any individual experiments. I was there on a shuttle. I transferred over 6 tons of equipment, including a lot of science that was used on the space station, and also helped with completing kind of a closed loop cycle for the space station where we're able to take our urine and recycle it, but also use it to break down through electrolysis, of course, for making oxygen, and then combining it in a sabatier with the carbon dioxide that we breathe out, and creating more water, and of course the methane is vented overboard. So that technology in itself has been able to allow our space station to work for longer, and our crew helped bring up the sabatier to close out that loop. And then we were able to do spacewalks that help allow the space station remain cooled so that all the equipment and science that's on the outside of the vehicle is taken care of and can continue on. And so that is what I have to say for you today. And again, thank you so much. And I really look forward to hearing about this winner who is moving on. But like I said, I really feel truly that each of you that participated has significantly contributed and is a winner in just being here today. Think about, there's been many that didn't even make it into this room. So thank you very much.
We are in the finale, and it is my pleasure to invite here the jury chairman, Professor Bužek, also to invite here the president of the Falling Walls Slovakia, with Professor Moco, and the representative of the German Academic Exchange, Mr. Christian Alrep, to announce the results.
I assume at this point all of you expect me to say, and the winner is, which probably I will use, and I plan to use exactly the same words as Dorothy did. Essentially, winners are all that participated in this competition. That is really fantastic. You have done a great job, really great job, being able to present your results to big audience in English, essentially to compare the quality, and we had a chance to compare the quality of those presentations, and we had, by the rules of the game, to select some of the winners. That was so tough, and therefore essentially our discussion of the jury was probably longer than originally expected. We thought our job would be easier, but it was very tough. But anyway, we had to do that. These are the rules of the game, and we had to select 3 finalists, 3 winners, in the order. So the first who won the whole competition will travel to Berlin. And I hope I have it correct. I'm supposed to announce the winners, the 3 winners in reversed order. So the first will be the third, second, and so on and so forth. You understand. So the last one, okay, so I hope that the tension is building here. Anyway, so I'm going to really to make the announcement. So the third place, the jury decided would go to Maciej Mikulski for Breaking Walls of Application Design. Second place is taken by Stefan Tkáč for breaking the wall of hydroturbine rotor stabilization. He's the boss. He's the boss. He's the boss.
On prvý šejkuje rukou.
And now, according to the jury, the winner of the whole competition in Falling Walls Lab Slovakia is Stanislav Hrivňak for his presentation of breaking the wall of small animal imaging. So, according to the rules of the game, Stanislav Hrivňak will go to Germany. Here's his ticket to Berlin, thanks to DAAD. Thanks a lot for really making this contribution.
I would just like to add a couple words. The German Academic Exchange Service is a state institution for the international scientific exchange, and we have 400 million euros every year in the budget for this exchanges, and like 70,000 scholarships, and this is one of the scholarship. And it is one flight to Berlin and 3 nights in Berlin, and the winner can attend the big conference, and afterwards he has 2 nights more in Berlin. I guess you will find some places. I heard there are 3,000 events every night. Have a good time. Good luck.
So thank you for the words of representative of DAD. My final comment maybe, and then the president will close the whole meeting. Please remember, next year there will be Falling Walls Lab Slovakia, the 2nd year, and I hope that many more will apply, and we will really be able to present the best we can produce here in Slovakia in 2018 in Berlin. Thanks a lot.
So, let me say something in the final part of our exciting event. First of all, congratulations to all participants, and to say it very simply, in this difficult, hard world, it is not enough to be Einstein, you have to be also first-class performer and actor. Because if you have great idea and if you do not sell it properly, then people not ready at your level to understand your topic can appreciate this. But this has been known. So this certainly contributed to our hard-making decision. I would like to thank, first of all, rectors of the universities and president of academy for their great support and understanding. Of course, they made it possible. Then I would like to thank you, members of the jury, and first of all, president of the jury, Professor Bužek. Without the jury, we wouldn't have winner, despite the greatness of all ideas. And now I would like to call 2 Marians, Dr. Marian Holienka, project manager, and his assistant, Magister Marian Sabo, because all really, come here please, Marian, because without their effort and their very efficient logistic, we would not be here today. So thank you, Marians, very much for your contribution. Vice rector from the same faculty. I thank you all who came here because really it was for us difficult to predict what will happen, whether we will succeed or not. As Professor Bužek already said, we were amongst the ambassador, we were amongst the most successful countries in invoking interest from our young, talented researchers, which is very good, and it was very positively quoted in Berlin. I invite you all for networking and reception outside this hall. Thank you very much.